Okay guys, last time we left off with our lipids. We finished lipids. So we're done with carbohydrates, we are done with lipids. Okay. What I need you guys to know for carbohydrates and lipids, again, make sure you know your monomers, make sure you know your polymers. Keep in mind with your condensation, dehydration, and hydrolysis reactions, how do they apply to the monomers and the polymers of our carbohydrates, of our lipids. So make sure you guys know, like when you're going from a monomer to a polymer, it's a dehydration or a condensation reaction. So if we are going from a monosaccharide, the monomer of a carbohydrate, to a disaccharide, okay, the polymer of a carbohydrate, that's a condensation or a dehydration reaction. If we are going the other way, from a polysaccharide to a monosaccharide, that's a hydrolysis reaction, okay? And the same thing with lipids. If you're going from a fatty acid to a triglyceride, that is a dehydration or condensation reaction. Go the opposite way. Take a triglyceride and break it apart into fatty acids. Well, then that's a hydrolysis reaction. So try to apply those terms, guys, all right, when you're going through this. We're going to go over proteins today, things like all these meats, cheeses, eggs, nuts, all these things, they contain proteins. You guys know proteins make you, strong, lovely. But our body actually naturally makes proteins as well. So you don't have to always think like bodybuilders up in a gym, you know, oh, bro, like I'm all done lifting, bro. Like I gotta go take some protein. I don't know why they sound like surfers, but whatever. So here is going to be our structure, our elements in our proteins. Now, I'm gonna put them somewhere else on our chart because we are going to fill up that structure box for our protein. So we have carbon, hydrogen, oxygen, nitrogen, and sulfur sometimes. Okay, not all the time. It's like A, E, I, O, U, and sometimes Y are our vowels. It's like hydrogen, carbon, oxygen, nitrogen, and sometimes sulfur. But anyway, our amino acid is going to be our monomer of our protein. So let's fill a little bit of the chart out now. Not too much, just a little. That's not the right one. Here it is. Okay. So let's go to we did carbohydrates, we did lipids. You guys know those are all carbon, hydrogen, and oxygen only. Let's go down to our protein here. And so our protein structure, we're going to fill out in a little bit, but what I'm going to do is right under proteins, I'm going to write carbon, hydrogen, oxygen, nitrogen, and sometimes sulfur. So if you remember, it was carbon, hydrogen, and oxygen up here, but not so much oxygen. And then for carbohydrates, it was carbon, hydrogen, and oxygen. So these differentiate from our lipids and our carbohydrates because we have nitrogen and sulfur in there now. So go ahead, write that one down on your papers, just so you guys know the difference between what elements are, in there. All right. So let me erase these. Boom, boom, and boom. So we're going to go over to our monomer of our proteins, and that is an amino acid. Okay, amino acid. All right. Okay, so in our proteins, guys, the amino acids, these are our monomers. There are 20 of them. Here are a few of our proteins, our amino acids here. We have some that are nonpolar, like the ones up here. We have some that are polar. Do you have to know all these? No. If you take biology in college, do you have to know all these? Yes. All right. So yeah, you'll have to know all the individual amino acids. And like I said, there's 20 of them altogether. Okay. So here is the structure of an amino acid. I ain't going to go through this with you guys. Let's do the structure instead, the picture of the structure, because that's going to make more sense. Now, what I would do here, guys, is I'm going to write this in. I'm going to write the structure here, and I'm going to enlarge my box so I can write everything I need to write in here. Don't write anything right now, guys. What I think you should do, especially if you're doing this digitally, I'm going to draw it, take a screenshot of it after I'm done drawing it, paste it in your document. I think that's the best way to do it. And I'm going to color code this very nicely for you. So we are going to start off with a central, lovely, wonderful carbon here in the middle. Coming off of that carbon, we are going to have one element. That element is hydrogen. Cool. Lovely hydrogen there. Okay. On one side, we are going to have an amino group. So I will color code this for you. This is our amino group. There you go. On the other side, we are going to have a carboxyl group. Okay, there you go. So if you're looking here, I got my central carbon, 1, 2, 3 bonds. You guys know carbon always has to form 4 bonds. So I need something else. I got an amino group on one side. I got a carboxyl group on the other side. I got a hydrogen coming off of it. My last part is going to be up here, and that's R. Look at the periodic table. Okay, go ahead. You looked. Yeah, we're done. I will tell you the answer. There is no R. The R is the variable group. There is no R element on the periodic table. Okay. So, how many variable groups are there? There's 20. Now this structure is our amino acid. Why is it called an amino acid? Well, we got an amino group on one side. We got a carboxyl group on the other side. Together, the amino group, boom, that's in the name. Carboxyl group makes it acidic, so that's why it's called an acid. All right, that is our amino acid there. All right. Come on, lights. It's dark in here. There we go. That was a close one. Sometimes it's scary when it's dark outside or inside. Yeah. All right. Okay, so that is it. I'm writing in the box, guys. I'm done. I would take a screenshot of that, paste it into your document, okay, because making this would be very difficult. All right. And now, if we look at the slide here, AA is amino acid, guys. Okay, the amino acid consists of a central carbon, hydrogen atom, R group, carboxyl group, amino group. Boom. We just drew all that out, and it makes a lot more sense than that. There's another picture. If you like that one better, boom. Make a little screenshot of that and paste it into your graphic organizer for the macromolecules. Either one works, guys. You like this one better, take a screenshot of it. You like mine better just because, you know, I drew it and it's fantastical? Draw, you know, put that one on there. Whatever you guys want. All right, moving on. That was a lot. We need some minions for applause. Yeah, there we go. I like the minions. All right. So we have our amino group on one side. We have our carboxyl group. On the other side, our central carbon, hydrogen atom, the red, that red color on top, that is our group. And you guys can see that it's going to be different for each one. I just wanted to show you guys the difference between our R groups. One has the H, one has CH3, okay? Just different R groups there. So now let's get into our polymers. So if we start to put our monomers together, we are going to, let me move myself out of the text here. We are going to have dipeptides and polypeptides. They did the same thing with this as they did with our carbohydrates. If you remember, for carbohydrates, we had, you know, our monosaccharide. Put two of them together, we get a disaccharide. Put more than two of them together, just call it a polypeptide or polysaccharide. It didn't matter how many there were after two. It was all polysaccharide. They did the same exact thing for proteins. One monomer is an amino acid. Two, not as nice because it was like monosaccharide to disaccharide, so it's a completely different name here. But they have a dipeptide for that. Okay, so sorry guys. Give me like 2 minutes, okay? So we have a amino acid. Put two amino acids together, we get a dipeptide. If we put more than that together, we get a polypeptide. So let's put that in our organizer here. So I'm going to go all the way over to our end here for our polymer of proteins, and we are going to put dipeptide, which is 2, or polypeptide, which is more than 2. And these are with respect to amino acids. All right. So go ahead, copy that one down, guys. Okay, cool. Okay, the last thing we got to go over, guys, is protein folding. So what's going to happen here is our proteins are going to get bonded together, forming our dipeptides or our polypeptides. And then what's going to happen is after we have this long chain, those R groups are going to combine together through hydrogen bonding. And we have all these different shapes. We don't have to worry about them. You don't need to know them right now. But in the end, it's going to form this globular protein. It's going to fold. Our nice long chain of proteins folds up. That way we can store it nicely inside of our bodies. All right, guys, we're going to do a little work with enzymes tomorrow. And have a good day. Review your charts tonight just to get familiar with them. And I'll see you tomorrow.